Usually when we talk about a medical diagnosis, there's this expectation of precision. It feels, I don't know, almost like engineering.
Right, yeah, like it's a mechanical problem you can just point to.
Exactly. Like you break your arm, the x-ray shows that jagged white line on the glowing screen, and the doctor just points a pen at it and says, there it is. It's a completely binary state. Broken or not broken.
It is really comforting to have that visual confirmation. I mean, we inherently like biological problems to be visible, localized, and easily categorized into a neat little box.
Right, we want to see the bad guy. But then you step into the world of neurodevelopment or neurotrauma or autoimmune diseases, and suddenly that x-ray machine is just useless.
Oh, completely useless. You're looking at a diagnostic landscape that is, quite honestly, incredibly murky.
Yeah, because the damage is microscopic, it's systemic, and often it's completely invisible until the symptoms are just overwhelmingly obvious.
It's the absolute definition of diagnostic muddy waters. I mean, the central nervous system just doesn't broadcast its failures the way a skeletal system does.
It really doesn't. And today, for you listening, we are wading right into the absolute deepest end of those waters. Welcome to the deep dive.
Yeah, this is a heavy one, but an important one.
Really important, because today we're looking at a disease that doesn't just physically damage the body. It fundamentally hacks the nervous system from the inside out. We are taking a PhD-level deep dive into Canine Distemper virus, or CDV.
Right, and for anyone who has ever raised a puppy or, worked in veterinary medicine, the word distemper carries a very specific, very heavy dread.
Yeah, it's a nightmare.
It really is. It's a devastating, highly contagious disease.
It is, but we're looking at this today not just as a veterinary tragedy. We're looking at it because the specific way this virus invades the brain and causes permanent neurological damage.
Specifically, a condition called demyelinating leukoencephalitis.
Yeah.
Right, that condition. It acts as an incredible, naturally occurring mirror for multiple sclerosis, or MS, in humans.
The pathological parallels between the two are just striking. I mean, tracing all the way down to the cellular and molecular levels.
It's wild how similar they are. But our mission for this deep dive goes beyond just, admiring the pathology. The ultimate goal today is to explore a breakthrough clinical study titled Epidural Cell Therapy Reduces Neurological Sequelae Severity in Canine Distemper Survivors.
Which is a brilliant paper. Truly groundbreaking stuff.
Yeah, we are basically starting with a microscopic detective story, like a literal home invasion at the cellular level.
And we're going to end with a piece of cutting-edge regenerative medicine that might just rewrite the rule book for how we treat irreversible brain damage.
Okay, let's unpack this. Because before we can understand the cure, we have to deeply understand the invader. What exactly are we dealing with here?
Well, canine distemper virus is an enveloped negative-sense RNA virus. It belongs to the Paramyxoviridae family and specifically sits within the genus Morbillivirus.
Okay, I want to pause right there on negative sense RNA, because that sounds like something straight out of a genetics textbook.
It is a bit dense, yeah.
Does negative sense mean the virus's genetic code is essentially written backwards?
That is actually a really functional way to think about it.
Yeah.
So in biology, human cells, or dog cells in this case, use positive sense RNA as the direct blueprint to build proteins.
Like reading an instruction manual.
Exactly. The cellular machinery, the ribosomes, they can read positive sense RNA like a book. But CDV brings negative sense RNA. It's a mirror image. The host cell's ribosomes look at it and they just can't read it at all.
So the virus brings a blueprint that the factory workers literally cannot understand. How does it ever do anything then?
It has to bring its own translator. The virus actually carries a specific enzyme called an RNA-dependent RNA polymerase.
Wow. Okay, so it brings its own decoding machine.
Right. The moment the virus enters the cell, this enzyme immediately gets to work transcribing the negative sense strand into a readable positive sense strand.
Mm-hmm.
Only then can the host cell actually start manufacturing viral proteins.
It's a whole extra step just to get started.
It is, but it's a hallmark of this entire family of viruses.
And this Morbillivirus genus, it has some pretty infamous relatives, right?
Very infamous. I mean, it's closely related to the human measles virus. In fact, they share a common viral ancestor.
That makes sense given the similarities we're going to talk about.
Yeah. It's also related to rinderpest, which is a now eradicated cattle plague that used to cause absolute famine. Mm-hmm. But CDV primarily infects carnivores. Dogs are the primary host we think of course.
But not just dogs, right?
No, not at all. It also sweeps through populations of ferrets, raccoons, lions, and even marine mammals like seals.
Man, that's broad. I'm trying to visualize the initial infection here. Like, is a dog getting this from a bite like rabies? Or is it more like a common cold?
It's respiratory. So transmission is almost exclusively via aerosol inhalation.
Just breathing it in?
Yeah. An infected animal sheds the virus in respiratory droplets, and another dog just breathes it in. The virus enters the upper respiratory tract. And once inside, it doesn't just stay in the lungs.
It goes everywhere.
It's incredibly versatile. It targets a really wide array of cell types. Epithelial cells lining the organs, mesenchymal cells forming connective tissues, neuroendocrine cells, and crucially, hematopoietic cells.
Okay, hematopoietic cells, let's bring that down. Hemat meaning blood. So these are the stem cells and their descendants that make up the entire blood and immune system.
Yes, exactly. The virus makes a beeline for the lymphatic system. It replicates heavily in the tonsils, the lymph nodes, the spleen, the thymus.
Wow, so it goes straight for the defense headquarters.
Right. And here's the truly devastating mechanism of the acute phase. It causes profound, long-lasting immunosuppression.
Just turns the immune system off.
It effectively turns off the body's defense systems by rapidly depleting CD4 plus T cells.
Okay, the CD4 plus T cells. Let's make sure we have the hierarchy of the immune system straight here. Those are the helper T cells, right?
Correct. They are the helpers.
So they act like the battlefield generals. They don't necessarily kill the virus directly themselves, but they coordinate the entire attack, like telling the B cells to make antibodies and directing the killer T cells where to go.
That is the exact role of the CD4 plus cells. Without them, the immune system is completely blind and disorganized. The virus likely depletes them by triggering a massive wave of apoptosis.
Apoptosis being programmed cell death. So the virus isn't just bursting the cells open. It's hacking their internal software and forcing the generals to essentially commit suicide.
Exactly. It's a highly regulated systemic die-off. And what makes this phase even more complex is that the CD8 plus T cells, which are the cytotoxic killer cells,
the actual foot soldiers doing the hand-to-hand combat?
Right. Those foot soldiers are much less affected by this initial wave of apoptosis.
That's bizarre. So we have an immune system where all the generals are dead, but the heavily armed foot soldiers are still wandering around the lymphatic tissues without any orders or coordination.
The clinical result is absolute vulnerability. I mean, if we connect this to the broader context of viral pathology, this severe, rapid immune depletion is virtually identical to what we observe in human measles.
Which is why measles used to be so deadly before vaccines.
Exactly. It leaves the host, whether it's a puppy or a human child, incredibly exposed to secondary infections. During the acute phase of distemper, it's very often not the virus itself that actually kills the animal.
Wait, really? What kills them then?
It's the opportunistic bacterial pneumonia or fungal infections or even secondary gastrointestinal parasites that just sweep in entirely unchecked while the immune system is offline.
So the virus essentially clears the runway for everything else to land.
Right. It opens the gates.
Okay, so the dog breathes it in, the virus takes out the generals, crashes the immune system, and starts spreading systemically. But our focus today is the brain.
Yes. The neurological aspect.
And the brain is legendary for its security system, right? The blood-brain barrier. Historically, I've always heard it described like a strict VIP bouncer outside an exclusive club.
It's a common analogy, yeah.
But I feel like that metaphor really doesn't do justice to the actual biology. How physical is this barrier?
Well, a bouncer implies someone just standing there checking a list at a door. The blood-brain barrier is much more akin to a high-security biological clean room airlock.
An airlock. Okay, I like that better.
The blood vessels in the brain are lined with endothelial cells, just like everywhere else in the body. But in the brain, these endothelial cells are fused together by what we call tight junctions.
Tight junctions, so they are literally stitched together.
Stitched together so seamlessly that practically nothing water-soluble can simply squeeze between them. And additionally, you have the foot processes of astrocytes.
Those are the star-shaped brain cells, right?
Yes. They wrap tightly around the outside of the blood vessels, adding a second physical and chemical wall.
Wow, okay.
To get into the brain tissue, a molecule or a pathogen generally has to be actively smuggled through the body of the endothelial cell itself.
So a virus floating freely in the bloodstream is just going to bounce right off the walls of this clean room airlock? How does canine distemper sneak past a system designed specifically to keep pathogens out?
It utilizes two distinct, really insidious methods to breach the central nervous system, or CNS. The first is a classic Trojan horse strategy.
Okay, a Trojan horse. How does that work here?
Because the virus infects mononuclear cells, which are circulating white blood cells like monocytes and lymphocytes,
It essentially hides inside them.
Oh, I see.
And immune cells are authorized to pass through the blood-brain barrier under certain conditions.
Yep.
They have the right chemical keys to be allowed through that airlock.
So the infected immune cell gets clearance to enter the brain, completely unaware that it's carrying this viral payload inside it.
Exactly. The virus just hitches a ride right past the tight junctions inside a legally entering cell.
That is terrifyingly efficient. What's the second method?
The second route bypasses the blood vessels in the brain parenchyma entirely and goes through the plumbing system. The virus circulates in the cerebrospinal fluid, the CSF.
Okay, so the fluid that bathes the brain and spinal cord.
Right. Deep inside the brain are hollow cavities called ventricles, which are filled with this CSF. And these ventricles are lined with a layer of cells called the ependymal lining.
So it attacks the lining.
Yeah, the virus in the CSF directly infects and fuses with these ependymal cells. It uses the lining of the brain's internal swimming pool as a beachhead.
Wow.
This is why when veterinary pathologists examine the brains of dogs that have died from early-stage distemper, they frequently find the initial inflammatory lesions clustered right around those ventricles and in the subvendible areas just beneath the surface of the brain.
So the invasion starts from the fluid borders and works its way inward. Okay, so the virus is now officially inside the central nervous system. This brings us to the acute phase of the neurological invasion. What exactly is the virus doing to the infrastructure of the brain once it establishes these beachheads?
This early CNS invasion typically occurs around 3 weeks post-infection. The timeline here is critical. Remember the state of the dog's immune system at 3 weeks.
Right, the CD4 plus T cells are still wiped out. The dog is severely immunosuppressed.
Exactly. Because of that systemic immunosuppression, there is very little inflammatory response happening in the brain at this stage. There are no massive waves of immune cells rushing in. It's a completely silent invasion.
Creepy. So what does it do while no one is watching?
The virus initially sets up shop by infecting astrocytes.
Those are the star-shaped cells you mentioned earlier that wrap their feet around the blood vessels?
Yes. Astrocytes are crucial for maintaining the chemical environment of the brain and supporting that blood-brain barrier. But while infecting astrocytes is disruptive, the truly critical damage, the pathology that leads to demyelinating leukoencephalitis, involves the infection of a completely different cell type, the oligodendrocytes.
Oligodendrocytes. Okay, let's dissect that word. Oligo meaning few, dendro meaning branches, cyte meaning cell, the cell with a few branches.
That's the literal translation, yeah.
But what they actually do is far more important than their shape, right? These are the cells that manufacture myelin.
They are the insulation manufacturers.
Yeah.
The brain is an electrical organ, communicating via electrical impulses traveling down long, thin nerve fibers called axons. If those axons are bare wires, the electrical signal leaks out, degrades, and travels incredibly slowly.
Like trying to send a text message over an uninsulated copper wire lying in a puddle of water.
That is a perfect analogy. To prevent that, the oligodendrocyte extends its branches and physically wraps layers and layers of fatty tissue, the myelin, around the axon.
So it's basically electrical tape.
Yes, this myelin sheath acts as electrical tape. It allows the nerve impulse to jump rapidly down the axon, ensuring fast, efficient communication. Without myelin, the neurological signals short-circuit, misfire, or just fail to reach their destination entirely.
Which manifests as paralysis, twitching, blindness, all the clinical signs of neurological failure.
Exactly.
Now, I understand how a virus normally kills a cell. It gets in, forces the cell to print 1 million copies of the virus, and the cell bursts like a balloon.
The classic cytolytic infection, yes.
But when I was looking through the sources, the pathology here gets incredibly strange. The virus doesn't just slaughter these oligodendrocytes in a traditional explosive way, does it?
No, it doesn't. And this is where the cellular biology gets really fascinating. The virus induces what researchers call a restricted infection.
Restricted in what sense? Like, is the virus trapped?
It's restricted in its life cycle. When virologists take tissue samples of these early brain lesions and look at the oligodendrocytes under incredibly high resolution, they find that viral transcription is actively occurring.
Okay, meaning?
The viral mRNA is clearly present inside the cytoplasm of the oligodendrocyte, but there is a complete absence of translation.
Oh, wait. Let me make sure the listener and I are totally clear on the distinction here. Transcription is the first step. It's when the viral enzyme reads the negative sense RNA and writes a readable positive sense copy, the mRNA messenger. And translation is the second step, where the cell's actual factory equipment, the ribosomes, read that mRNA messenger and use it to build the physical 3D proteins that make up the new viruses.
That's the exact sequence.
Yeah.
So in these oligodendrocytes, the viral blueprint,
the mRNA, is floating around inside the cell. But the cellular machinery never actually reads it.
Never.
Never. It never builds the viral proteins. The oligodendrocyte does not become a virus-producing factory.
So the cell just ignores the viral blueprint? Honestly, that sounds like a victory for the dog. The cell isn't making new viruses.
You'd think so. It would be a victory, except the presence of this viral mRNA completely derails the cell's normal operations.
How so?
The cell seems to recognize that its internal environment has been compromised. The consequences of this restricted infection are dire. There's a massive systemic downregulation of the oligodendrocyte's own normal genetic functions.
It just shuts down.
Specifically, researchers see a drastic, sudden drop in the transcription of myelin-specific genes.
Can you give an example of one of those genes?
The primary one is the proteolipid protein gene, or PLP. PLP is one of the foundational building blocks of the myelin sheath. When the cell stops transcribing PLP, it stops making myelin.
Man, okay. I'm picturing a highly efficient manufacturing plant. The workers, the ribosomes, and the cellular machinery suddenly receive these hacked alien blueprints dropped onto the factory floor.
Right.
They don't actually build the alien machines. The viral proteins aren't made. But the sheer presence of this foreign code triggers a factory-wide lockdown.
That's exactly it.
The assembly line completely freezes. They stop building their normal product, the myelin. And myelin isn't a one-and-done product, right? It requires constant upkeep.
Yes, constant metabolic maintenance.
So if the factory stops producing new myelin, the existing insulation slowly starts to degrade and collapse.
It's a profound metabolic dysfunction. The cell doesn't die immediately, but it completely abandons its primary job. And the failure to maintain the myelin sheath leads to a slow, insidious demyelination.
That's brutal.
It is. However, we have to add another layer of complexity to this pathology because the latest neuropathological research points strongly to the inside-out model of demyelination.
Inside out. Meaning the damage isn't actually starting with the myelin on the outside of the wire.
Right. For decades, the prevailing assumption in veterinary medicine, and in MS research as well, was that the primary pathological event was simply the myelin degrading off the axon. An outside-in destruction.
Which makes sense based on what we just talked about.
It does, but advanced imaging and molecular analysis now provide strong evidence that early axonal damage actually precedes the loss of the myelin.
Wait, how does the virus damage the wire itself if it's targeting the insulation factory?
The virus, either directly or through the metabolic chaos it causes in the surrounding cells, disturbs the axonal cytoskeleton.
The cytoskeleton being the internal structural scaffolding of nerve fiber.
Yes. Axons aren't just empty tubes. They have a complex internal network of microtubules and neurofilaments.
It's like little highways inside the nerve.
Exactly like highways. They transport nutrients, proteins, and mitochondria from the main cell body all the way down to the nerve endings. The virus disrupts these transport mechanisms.
Oh, wow.
The structural scaffolding begins to break down. The axon itself begins to suffer metabolic starvation.
So the copper wire itself starts to rot from the inside out, and then the insulation falls off because the wire underneath it is dying.
Precisely. The oligodendrocyte, which is intimately wrapped around that axon, senses that the axon is failing. Deprived of the healthy electrical and chemical signals from a functional axon, the oligodendrocyte starts to undergo dystrophy. It withdraws its processes.
That's a double whammy.
It really is. So we have a dual mechanism of destruction. The restricted infection, halting myelin production, and the inside-out rotting of the axonal wire. And while all this silent, cellular-level destruction is happening, another player in the brain is waking up, the microglia.
Okay, the microglia. These are the brain's resident built-in immune cells, right? Because the peripheral immune system, the T cells and B cells in the blood, are kept out by the blood-brain barrier. So the brain has to have its own local security force.
Exactly. They're essentially macrophages that permanently reside in the central nervous system. And even though the dog's systemic immune system is currently wiped out by the virus, these local microglia begin to react.
They realize something's wrong.
They sense the viral RNA. They sense the metabolic distress of the oligodendrocytes and the degrading axons.
How do they react? What do they physically do?
They undergo activation. They physically change shape, becoming more amoeboid, allowing them to move toward the damage.
Like they're getting ready for a fight.
Crucially, they begin to upregulate major histocompatibility complex class II molecules, or MHC2, on their surfaces.
MHC2, those are the display windows, right? The molecules that cells use to hold up pieces of a pathogen to show the rest of the immune system what they found.
Yes, they are preparing to present antigens.
They also start producing early pro-inflammatory cytokines, which are chemical warning signals. The microglia are essentially pulling the fire alarm, lighting flares, and setting the stage for a massive, catastrophic response once the rest of the body's immune system finally comes back online.
Which leads us to what feels like the central tragic irony of this entire disease narrative. I mean, the dog has been severely immunosuppressed. The virus has silently invaded the brain, initiated a restricted infection in the oligodendrocytes, halted myelin production, and started rotting the axons from the inside out.
Yes.
But eventually, a dog that survives the initial respiratory infection is going to fight back. The immune system has to bounce back at some point. When does that happen?
In the dogs that survive that acute systemic phase, the peripheral lymphoid tissues begin to repopulate around 6 to 7 weeks post-infection.
Okay.
The bone marrow ramps up production, the thymus recovers, the immune system finally comes back online.
So logically, as a layperson looking at this, if the immune system bounces back, the dog should start getting better.
Right.
The cavalry has finally arrived. They should flood into the brain, hunt down the virus, clear the infection, and the brain should heal.
It is the most logical assumption to make. You would expect recovery. But biology is rarely that straightforward. What actually happens is the exact opposite.
The opposite.
The return of the immune system heralds the chronic inflammatory phase of the disease, and it is absolutely devastating.
Why? What do they do?
Well, as the immune system repopulates, a massive flood of newly minted CD4 plus T cells, CD8 plus T cells, and B cells arrive at the blood-brain barrier. Remember those flares the microglia let?
Right, the chemical signals pulling the alarm.
They guide these cells straight into the brain.
So the generals and the foot soldiers are back from the dead, and they are furious.
They are highly activated. The B cells cross into the brain parenchyma and immediately begin producing massive amounts of intrathecal antibody.
Intrathecal, meaning they are synthesizing these antibodies right there inside the cerebrospinal fluid and brain tissue, rather than producing them in the blood and hoping they cross over.
Yes. It's a localized, intense, humoral immune response. And we have to give the immune system credit where it is due. This intense cellular and humoral attack actually does what it's biologically programmed to do. It successfully clears the actively replicating virus from those early inflammatory lesions.
Wait, it clears it? But based on how you describe this as a tragic irony, I am guessing the cure is worse than the disease.
The cure is catastrophic. The process of clearing the virus causes incredible collateral damage to the delicate architecture of the brain tissue. In neuropathology, we term this phenomenon bystander demyelination.
Bystander demyelination, like innocent civilians caught in a massive artillery barrage. The immune system is carpet bombing the city to get to a few hidden insurgents.
That is a very accurate, albeit grim, way to picture it. To understand exactly how this friendly fire destroys the brain, we have to look deeply at the cytokine storm. You recall cytokines are the chemical messenger proteins that immune cells use to communicate, direct traffic, and escalate or de-escalate an immune response.
Right. They are the radio signals of the immune system.
In this chronic phase of distemper, the orchestration of these signals becomes completely dysregulated. There's a massive sustained upregulation of pro-inflammatory cytokines. We're talking about interleukin-1, interleukin-6, interleukin-12, and profoundly tumor necrosis factor alpha, or TNF-alpha.
Let's break those down because an interleukin just sounds like generic science jargon. What is interleukin-6 actually doing in the tissue?
Think of interleukin-6 and interleukin-1 like biological flare guns that also act as a siren.
Okay.
When released into the tissue, they bind to receptors on other immune cells and trigger them to become hyperactive, to multiply, and to release even more destructive enzymes. They amplify the panic.
And tumor necrosis factor alpha, that sounds particularly aggressive.
TNF-alpha is a primary driver of the destruction. One of its crucial roles in this pathology is promoting leukocyte extravasation.
Extravasation, the process of getting white blood cells out of the blood vessel and into the tissue.
Yes. TNF-alpha acts on the endothelial cells of the blood vessels. It makes them sticky, causing white blood cells to roll and adhere to the vessel wall.
Okay.
Then it chemically pries open those tight junctions we talked about earlier, the high-security airlock.
It just forces the doors open.
It forces the endothelial cells to contract, creating physical gaps that allow even more immune cells and inflammatory fluid to rush from the blood into the brain.
It is actively dismantling the blood-brain barrier from the inside to let more troops in. So the pro-inflammatory signals are screaming at maximum volume. Are there no chemical signals trying to turn the volume down?
That is the crux of the dysregulation. The immunomodulatory cytokines, the signals that are biologically designed to act as the braking system, to tell the immune system to calm down and begin tissue repair.
Things like interleukin-10.
Right, so cytokines like interleukin-10 and transforming growth factor beta, or TGF-beta, they barely register. They remain at baseline levels.
The gas pedal is bolted to the floor and the brake lines are completely severed. The brain is quite literally on fire with inflammation.
It's an unchecked positive feedback loop. And this inflammatory fire is physically fueled by a critical imbalance between two specific types of proteins operating in the background: matrix metalloproteinases, or MMPs, and their inhibitors, TIMPs.
Okay, let's unpack that. Matrix metalloproteinase. I know the matrix refers to the extracellular matrix, which is this structural scaffolding, the biological glue that holds all the cells in an organ together. What do the MMPs do to it?
MMPs are specialized enzymes containing zinc or calcium ions. That's the metallo part. Their specific biological job is to cleave or slice apart the proteins that make up that extracellular matrix.
So they break down the glue.
Yes. In a healthy, functioning body, MMPs are essential. They're used for normal tissue remodeling, for wound healing, for clearing away dead cells so new ones can grow. They are precision surgical tools.
But in the middle of this cytokine storm, I'm guessing they aren't acting like precision tools anymore.
They are weaponized. The massive influx of inflammatory cytokines triggers a huge overproduction of MMPs, particularly MMP2 and MMP9, by the invading immune cells and the local microglia.
And what do they destroy?
These enzymes begin chewing indiscriminately through the extracellular matrix of the brain. They actively degrade the structural proteins of the blood-brain barrier, exacerbating the leakiness that TNF-alpha started. Furthermore, MMPs can directly degrade the proteins that make up the myelin sheath itself.
They're literally dissolving the physical structure of the brain. And you mentioned TIMPs. Are those the brakes for the MMPs?
Yes. TIMPs stands for tissue inhibitors of metalloproteinases. They are specialized proteins that bind directly to the active site of the MMP enzyme, shutting it down. There is also another inhibitor called REC. The body releases these to keep the MMPs strictly controlled.
So why don't they work here? Why aren't they stopping the destruction?
It's a numbers game. In the chronic phase of canine distemper, the sheer volume of active, destructive MMPs completely overwhelms the available TIMPs and RECK inhibitors.
There's just too many of them.
The balance is entirely shattered. The enzymes are free to dissolve the tissue, fueling severe progressive growth of the demyelinating lesions.
The matrix is dissolving, the blood barrier is wide open, the inflammation is raging unchecked. Where do the macrophages fit into this horrifying picture? I know they are the heavy eaters of the immune system, the cells that physically swallow debris and pathogens.
The macrophages are the primary executioners when it comes to bystander demyelination. Remember the massive amounts of intrathecal antibodies being produced by the B cells?
Yes, the localized antibodies hunting the virus.
When one of those antibodies encounters a piece of viral debris or an infected astrocyte near an oligodendrocyte, it binds to it. Macrophages are constantly patrolling this area. They have specific receptors on their surface called Fc receptors.
Fc receptors, what do?
The Fc receptor specifically recognizes and binds to the tail end of an antibody.
Okay, so it locks on.
So when a macrophage bumps into an antibody that is locked onto a target, the macrophage's Fc receptor binds to it. This binding event acts as a hardwired trigger. It commands the macrophage to immediately undergo what is known as a respiratory burst.
A respiratory burst. That sounds incredibly violent. What is actually happening biochemically?
It is a localized form of chemical warfare. The macrophage rapidly ramps up its oxygen consumption and uses an enzyme complex to convert that oxygen into highly reactive oxygen species, or free radicals, things like superoxide anions and hydrogen peroxide.
Whoa.
It spews these toxic chemicals out into the immediate surrounding tissue to utterly obliterate whatever the antibody was attached to.
It essentially bathes the area in biological bleach and peroxide to melt the pathogen.
Exactly. And here is the fatal tragedy of the oligodendrocyte. By their very biological nature, oligodendrocytes are incredibly rich in iron.
Iron? Why do they need iron?
They require large amounts of iron as a cofactor for the enzymes that synthesize myelin.
Oh, I see. And the macrophage is spraying reactive oxygen species everywhere.
You see the chemistry.
Yeah, that's not good.
The high concentrations of intracellular iron in the oligodendrocyte react violently with the oxygen radicals released by the macrophage. This triggers a massive cascade of oxidative stress, lipid peroxidation, and DNA damage.
So it's basically rusting them.
The oligodendrocyte is literally oxidized to death. It rusts and melts, simply because it was standing next to a piece of viral debris when the immune system decided to launch a respiratory burst.
That is the ultimate definition of friendly fire. The immune system is desperately trying to sterilize the brain to save it, and the very chemistry of that sterilization process is destroying the insulation factories.
It's tragic.
But wait, let's go back to something you said earlier.
Yeah.
You said this massive immune response successfully clears the virus from these active inflammatory lesions. If the virus is gone, shouldn't the immune system eventually pack up and go home? Is this entire chronic phase just a runaway autoimmune hangover that never stops?
Well, if the virus were truly completely gone, the inflammation might eventually subside, leaving only the scar tissue behind. But the canine distemper virus is not gone. It's merely adapting.
Adapting how?
Certain strains of CDV, notably the A7517 strain, have evolved the ability to establish deep, permanent persistence in the central nervous system. They go into a profound stealth mode.
Stealth mode? I am struggling to understand how a virus, which is just a tiny envelope of proteins and RNA, can possibly hide from a raging cytokine storm, thousands of macrophages doing respiratory bursts, and a sea of targeted antibodies. Where does it hide?
It doesn't hide in a location.
Right.
It hides by fundamentally altering its own biological behavior and how it interacts with the host cell. And to understand this, we have to look closely at the viral F protein, the fusion protein.
Okay, what does the normal unmutated fusion protein do during an acute infection?
Normally, the F protein is expressed on the surface of the viral envelope. It acts as the grappling hook and the blowtorch. When the virus bumps into a host cell, the F protein forces the viral envelope to fuse with the host cell membrane, allowing the viral RNA to spill inside.
That makes sense.
Furthermore, as the newly hijacked cell starts producing millions of new viral F proteins, those proteins get embedded in the host cell's own outer membrane.
So the infected cell is now covered in viral fusion hooks.
Yes. And if that infected cell bumps into a healthy neighboring cell, the F proteins force the two cells to fuse together, and then they fuse with another and another.
Like a blob.
They create a giant multinucleated zombie cell called a syncytium. Eventually, this massive syncytium becomes unstable, bursts apart, and releases millions of newly assembled viruses into the extracellular fluid to start the process over again.
That is a cytolytic infection. It bursts the cell open, and the virus particles float freely in the fluid between cells, which means they are completely exposed to all those antibodies and macrophages we just talked about.
Precisely. A cytolytic infection is loud. It attracts the immune system. But the persistent strains of CDD, the ones that drive the chronic neurological disease, show distinct genetic mutations in a specific area of the F protein called the pre-sequence.
What does a mutation in the pre-sequence do to the virus's behavior?
It severely limits the transport and expression of these fusogenic complexes on the surface of the host cell. The cell still has the viral RNA inside it. It's still technically infected, but it doesn't get covered in those fusion hooks.
So it stops fusing cells together into those giant syncytia.
It stops fusing them, and critically, it results in a non-cytolytic spread. The infected cells do not burst. They don't explode and shed millions of free-floating progeny viruses into the extracellular space.
Then how does the virus spread to the next cell? Does it just stay trapped in the one astrocyte forever?
No, it uses the incredibly dense, tightly packed physical architecture of the brain to its advantage. Brain cells, astrocytes, neurons, oligodendrocytes, they are in constant physical contact with each other, passing nutrients and signals. The virus exploits these direct cell-to-cell contacts. It quietly passes its genetic material, the viral ribonucleoprotein complexes, directly through the microscopic junctions between the cells.
It is moving entirely indoors. It never steps out into the extracellular fluid.
Exactly. Because it's not bursting cells open, and because the viral particles are never floating freely in the interstitial fluid, the virus is never exposed to the circulating antibodies.
That's brilliant in a horrible way.
It never triggers the massive alarms of the humoral immune system. It remains completely hidden inside the protective shell of the host cells, slowly, quietly passing its corrupted code from cell to cell, constantly driving a low-level, chronic relapsing immune response that slowly chews the brain to pieces over months and years.
Which leaves the dog with permanent, horrific, progressive neurological sequelae. We're talking about symptoms like myoclonus, which are those involuntary, rhythmic, repetitive muscle twitches that look almost like a continuous focal seizure.
Yes, myoclonus is very common.
They develop ataxia, meaning they completely lose coordination and balance. They develop tetraparesis, which is the progressive weakness and paralysis of all four limbs. For decades, essentially since veterinary medicine began, the consensus has been that once a dog reaches this chronic demyelinating phase, the damage to the central nervous system is irreversible.
The prognosis has historically been exceptionally grim. Euthanasia was often the most humane option. Palliative care, perhaps using anti-seizure medications or corticosteroids to try to dampen the inflammation, was truly all that could be offered.
Because you can't unmelt a cell.
Right. You cannot unmelt an oligodendrocyte or unrod an axon with a pill.
But here's where this entire conversation takes a massive pivot. Here's where it gets really interesting, because this is the core of the deep dive today. We are looking at a potential paradigm shift.
We really are.
We are diving into a paper published in Stem Cell Research and Therapy detailing a groundbreaking clinical study aimed not just at managing the symptoms, but actively rescuing these survivors using epidural cell therapy.
It's an incredibly bold, fascinating study. The researchers took 4 mongrel dogs, mixed breeds, that had survived the acute systemic phase of canine distemper. The selection criteria here are absolutely crucial.
Why? What was special about them?
These dogs were not newly infected. They had been presenting with chronic, progressive neurological signs, the severe myoclonus, the paralysis, the pronounced head tilts, for over 6 months.
6 months. That is an eternity in neurological decline.
It is. By 6 months, the acute cytokine storm is long gone. The damage is baked into the architecture of the brain. The glial scars have formed. The myelin is completely stripped away.
The disease state was deeply entrenched.
Exactly. And importantly, the researchers confirmed through PCR testing that there was no active viremia present. The virus was no longer actively circulating or replicating in the blood. These dogs were dealing entirely with the chronic immunopathological aftermath, the viral persistence in the tissue, and the profound demyelination.
So dealing with dogs that conventional medicine would consider a lost cause, what exactly did the researchers do?
They treated the dogs with a specific type of stem cell called mesenchymal stromal cells, or MSCs. The dosage protocol was robust. 1 million cells per kilogram of the dog's body weight.
That's a lot of cells.
But the true innovation of the study was the delivery method. They didn't put them in an IV bag. They delivered them epidurally.
Into the spine.
And they didn't just do it as a one-off treatment. They administered 3 distinct doses, rigorously spaced 30 days apart.
Okay, there's a massive amount of biology to unpack there. Let's break this down piece by piece, starting with the cells themselves. Mesenchymal stromal cells.
Yes, the MSCs.
I have this pop culture image of a stem cell as a blank slate, a magical cellular brick that you can throw into a damaged organ and it magically turns into whatever cell is missing. Are these MSCs acting as replacement parts? Like, are they migrating into the dog's brain, looking around and physically transforming into brand new oligodendrocytes and neurons to patch the microscopic holes?
No. And it's vital to dispel that cellular brick myth.
Oh, really?
While it's true that MSCs can be coaxed to differentiate into various cell types in a highly controlled petri dish environment, in a living organism, and especially within the incredibly complex microenvironment of the central nervous system, they're simply not their primary mechanism of action.
So what are they doing?
They are absolutely acting as project managers, or perhaps more accurately, as microscopic field medics and foremen.
How do they manage the project without becoming part of the building?
They work through what we call paracrine effects.
Paracrine, meaning they don't have to touch the target cell. They secrete chemical signals into the local fluid to influence the behavior of the cells around them.
Precisely. They act as microscopic, highly responsive pharmacies. When they enter the damaged tissue, they analyze the chemical environment and begin reshaping the chronic inflammatory microenvironment.
Okay, give me an example of how that works in this case.
Let's go back to that positive feedback loop of destruction we discussed earlier. Or the microglia, the brain's resident immune cells that were stuck in a hyperactive pro-inflammatory state.
Yes, the cells lighting the flares and churning out the TNF-alpha and MMPs, the M1 phenotype.
Right. The MSCs enter that chaotic environment and immediately begin secreting specific immunomodulatory factors. Things like prostaglandin E2 and indoleamine 2,3 dioxygenase.
Okay, and what do those do?
These factors actively force a phenotypic shift in those surrounding microglia. They literally reprogram the local immune cells.
commanding them to transition away from the neurotoxic, pro-inflammatory M1 state and shifting them into the anti-inflammatory tissue-repairing M2 state.
So the MSCs walk into the middle of this microscopic riot. They don't throw punches. They just start handing out heavy tranquilizers and megaphones. They forcefully tell the immune system to stand down, drop the weapons, and start cleaning up the debris.
That is exactly step one. They quench the fire, but quenching the fire doesn't rebuild the house.
Right, the insulation is still gone.
Step two is active regeneration. The MSCs secrete incredibly potent molecules called neurotrophic factors.
Neurotrophic. Let's look at the root words. Neuro, referring to the nervous system, and trophic deriving from the Greek word for nourishment or growth. Brain food.
Yes, but highly specialized brain foods.
Yeah.
Specifically, the researchers highlight that MSCs secrete brain-derived neurotrophic factor, or BDNF, glial cell-derived neurotrophic factor, GDNF, and vascular endothelial growth factor, VEGF.
This is more alphabet soup. Let's ground these in physical reality. What do BDNF and GDNF actually do to a damaged, demyelinated nerve fiber?
BDNF and GDNF are absolutely crucial for neuronal survival in a toxic environment. But beyond survival, they actively promote synaptic plasticity.
Synaptic plasticity meaning the ability of the neurons to form new connections.
Right. If a specific pathway is destroyed by a demyelinating lesion, these factors help the surviving neurons sprout new dendrites and form new synapses to physically bypass the damaged area, restoring the circuit.
So they build detours.
Exactly. Furthermore, they stimulate axonal regeneration, helping those internal wires that were rotting from the inside out to repair their cytoskeletons.
That's huge.
And perhaps most vitally for distemper and MS, these factors stimulate the endogenous precursor cells that already exist in the brain.
Wait, endogenous precursor cells? You mean the brain has its own reserve pool of stem cells?
It does. They're called oligodendrocyte progenitor cells, or OPCs. They're sitting there in the tissue, but in a chronic inflammatory state, they're frozen. They can't mature.
The environment is too toxic.
Exactly. The BDNF and GDNF secreted by the MSCs signal these resident OPCs to finally wake up, migrate to the demyelinated axons, mature into fully functioning oligodendrocytes, and begin the painstaking process of wrapping new myelin sheaths around the bare wires.
They kickstart the remyelination process. And what about the VEGF? Vascular endothelial growth factor. Vascular means blood vessels.
Healing tissue requires massive amounts of energy. VEGF promotes angiogenesis, which is the physical growth of new, healthy blood vessel capillary networks into the damaged area,
To bring in supplies.
ensuring that all these newly active repairing cells get the heavy flow of oxygen and glucose they need to sustain the massive metabolic effort of rebuilding the brain.
That is just incredible. The MSCs aren't the bricks. They are the architects, the foremen, the riot police, and the supply chain managers all combined into one cell. They modulate the immune system, trigger the local stem cells, and build the supply roads.
They are doing all of it simultaneously.
But this brings me to a very practical clinical question. If these mesenchymal stromal cells are so incredibly smart and they secrete all these potent factors, why put a neurologically compromised dog under general anesthesia to perform a highly delicate epidural injection into the spine? Why not just hang an IV bag and infuse the million cells per kilo directly into a vein in the dog's leg? It is infinitely easier, cheaper, and less risky.
Intravenous delivery is undeniably easier. And historically, when stem cell therapies were first being conceptualized, 5e infusion was the most common route attempted for a myriad of diseases.
It makes intuitive sense. Just put it in the blood.
But the clinical results for neurological conditions were always incredibly inconsistent and often completely ineffective. And the reason comes back to the anatomy we discussed earlier, the blood-brain barrier.
The clean room airlock. Even though MSCs are therapeutic, they are still massive foreign cells. The endothelial tight junctions won't let them out of the bloodstream and into the brain tissue.
Most of them cannot cross. But the problem with IV delivery actually starts long before the cells even reach the blood vessels of the brain. When you inject millions of MSCs into a peripheral vein, that blood immediately travels back to the right side of the heart, and then it is pumped directly into the lungs to be oxygenated.
Right, the pulmonary circulation.
The capillary beds in the lungs are microscopic, incredibly narrow networks designed purely for gas exchange. MSCs, relatively speaking, are large, somewhat sticky cells. What happens is that the vast majority of these therapeutic cells simply get physically wedged and trapped in the tiny capillary networks of the lungs.
They just get stuck.
The ones that manage to squeeze through are often then trapped by the filtration systems of the liver and the spleen.
In pharmacology, we refer to this massive loss of therapeutic payload as the lung-liver trap.
So the rescue team gets stuck in traffic on the other side of the city before they even reach the highway to the brain.
Exactly. Dynamic MRI studies in rodent models, where researchers tag MSCs with magnetic contrast agents and watch them in real time, have shown devastatingly poor biodistribution. Less than 1% of intravenously infused MSCs actually managed to reach the brain parenchyma within 1 hour of infusion.
Wow. Less than 1%.
You are essentially throwing away 99% of your incredibly expensive, highly cultured therapeutic payload.
Okay, so doing an epidural completely bypasses all of that cardiovascular plumbing.
It does. An epidural injection places the cells directly into the epidural space, the fat-filled space just outside the dura mater, which is the tough, outermost membrane encasing the spinal cord and the brain. But placing the cells here, you allow direct, localized access to the central nervous system via the cerebrospinal fluid pathways and the local meningeal blood vessels. You avoid the lungs entirely.
But the epidural space runs the entire length of the spine and connects all the way up to the brain. It is a massive physical area. How do the MSCs know where to go once they're in there? The dog might have demyelinating lesions in its cerebellum or its brainstem or midway down its thoracic spinal cord.
Right.
If the cells just float around randomly, the odds of them landing exactly on the damaged tissue seem impossibly low.
This is where the biological elegance of MSCs truly shines, the homing mechanism. MSCs do not migrate randomly. They are highly modal cells that actively read chemical gradients in their environment.
They can track things.
Advanced studies using multimodal imaging, where researchers track the cells by labeling them with fluorescent quantum dots or tracking their metabolic activity using PE scans, show that these cells exhibit targeted migration. They literally follow a chemical breadcrumb trail.
What exactly are the breadcrumbs made of?
They are made of the very same inflammatory signals that are causing the damage. The astrocytes and microglia in the demyelinating lesions are pumping out chemokines and cytokines, screaming for help. MSCs possess highly specific surface receptors like CXCR4, which binds to a chemokine called SDF1, released by damaged tissue.
So the MSC has a physical antenna tuned specifically to the frequency of tissue damage.
Yes. When placed in the epidural space, the MSCs sense these chemical gradients diffusing out from the lesions. They actively migrate up the concentration gradient. Studies show rapid, targeted accumulation of MSCs directly within the CNS lesion areas, the specific patches of damaged white matter, often within a matter of hours.
That's incredibly fast.
They are guided directly to the site of the injury by the molecular SOS signals emitted by the injury itself.
The disease is literally broadcasting the GPS coordinates to its own cure. That is spectacularly elegant biology. So the theory is incredibly sound. The paracrine mechanism makes sense. The epidural delivery bypasses the lung trap. The homing mechanism ensures targeting.
Exactly.
But did this beautiful theory actually translate to clinical success for these 4 dogs? We have 4 dogs that have been suffering horrific neurological decline for over 6 months with permanent scarring. What actually happened after the injections?
Well, to measure the results objectively, rather than just relying on owner anecdotes, the veterinary researchers utilized an adapted application of a modified neurodisability scale, assigning the dogs a clinical score from 0 to 15 based on the severity of their symptoms. They paired this with a health-based quality of life questionnaire, scoring from 0 to 7.
So for the neurodisability scale, 15 would be catastrophic failure, and 0 would be completely normal neurological function. You want the disability score to go down and the quality of life score to go up.
Exactly. The researchers broke the neurodisability scale down into 5 distinct subcategories to pinpoint exactly what was healing: cerebellar function, postural abnormalities, cerebral function, ambulatory status, and myoclonus.
Okay, and they evaluated them throughout the process.
They evaluated the dogs at baseline and then systematically after each of the 3 epidural doses.
And the findings?
The findings were nothing short of remarkable for a disease considered untreatable. By the 4th evaluation, meaning after the full course of 3 doses had been administered and given time to act, complete clinical remission was achieved in 4 out of the 5 subcategories for almost all of the subjects.
Wait, complete remission, as in the clinical symptoms completely vanished. The veterinary neurologists couldn't detect them.
The scores for cerebellar function, which dictates coordination and fine motor control, dropped to 0. Postural abnormalities like the severe head tilts or the inability to right themselves when flipped over completely normalized to 0. Cerebral function, which includes their mental status, responsiveness, and cranial nerve reflexes, normalized to 0.
That's unbelievable. And what about walking?
Ambulatory status, their physical ability to stand up and walk, was actually the very first metric to show significant, measurable improvement.
Walking is such a massive threshold. If a paralyzed dog regains the ability to walk, their entire interaction with the world opens back up.
The ambulatory scores jumped significantly after just the first and second doses. By the end of the study, all but one dog had achieved complete remission in ambulatory status, moving from paralysis or severe tetraparesis back to functional walking.
That is undeniably life-changing. But you specifically said complete remission in 4 out of the 5 subcategories. What was the holdout? What didn't go to 0?
Myoclonus, the severe, involuntary, rhythmic muscle twitching that is the hallmark of chronic distemper.
Why is that? Why would the ataxia heal, the paralysis heal, but the twitching remain?
It proved to be the most stubborn, resistant symptom by a wide margin. Now, to be clear, the severity scores for myoclonus did significantly improve across the board for all the dogs over the course of the treatment.
So it got better, just not completely gone.
Right. The twitches became less violent, less frequent. But only one dog out of the 4 achieved full absolute remission, a score of 0 for myoclonus.
What makes myoclonus so much harder to cure than the other symptoms? Does it imply a different kind of damage?
Neurologists believe it likely reflects much deeper, more refractory neuronal damage. Myoclonus in distemper is often generated by central pattern generators located deep in the spinal cord or the brainstem.
Okay.
These are circuits of neurons that are supposed to be tightly regulated by inhibitory interneurons. The virus and the subsequent inflammation often permanently strip away or destroy those inhibitory controls.
So the brakes are gone.
Once that rhythmic, uncontrolled firing pattern is structurally established deep in the spinal cord, it is incredibly difficult to break the circuit and restore the inhibition.
It's like a short circuit that has actually melted the fuse box into a new configuration. It suggests that while 3 doses were amazing for the white matter tracks, treating severe entrenched myoclonus might require even more persistent long-term dosing.
Very likely, yes.
Which brings up the dosing schedule. You mentioned earlier that the study proved 3 doses were vastly superior to just doing 1.
The data provides undeniable proof of the necessity of repeated dosing. If you look closely at the timelines for cerebellar function, for instance, the clinicians noted minor improvements after dose 1, but it didn't completely normalize, it didn't drop to 0 until the 3rd evaluation time point.
So you have to keep going.
The data strongly suggests that persistence in dosing is vital for chronic demyelinating conditions.
Why is that biologically? Do the MSCs die off?
Yes. MSCs are not immortal in the host tissue. They arrive, they secrete their massive payload of paracrine factors, they modulate the local cells, and eventually, over the course of a few weeks, they're cleared away by the body.
So their effect is temporary.
A single injection of MSCs undoubtedly alters the environment temporarily, but a chronic disease state of 6 months is deeply entrenched. The local microglia will eventually start to revert to an M1 state.
It's like weeds growing back.
Exactly. It takes repeated waves of these project managers to continually suppress the inflammation, maintain the M2 shift, sustain the high levels of neurotrophic support, and buy enough time for the incredibly slow biological processes of axon repair and remyelination to actually take hold and complete the structural rebuild.
So if a well-meaning clinician tries a single epidural dose, sees minimal improvement after 2 weeks, and throws in the towel, declaring it a failure, they might be stopping the protocol right before the miracle actually happens.
That is a very fair, highly clinically relevant assessment based on these findings. The cumulative effect is what drives the structural healing.
It is just a marvel to step back and look at the holistic outcome here. We aren't just talking about a statistically significant drop in a clinical score on a piece of paper in a lab.
No, we're talking about real quality of life.
Yeah, the quality of life metrics skyrocketed across the board. These dogs became active again, they engaged with their environments, they regained their mobility and their autonomy.
They got their lives back.
They essentially got their lives back from a disease that for a century we thought was an absolute permanent death sentence for the nervous system.
It represents a profound, fundamental shift in the possibilities of veterinary neurology.
Okay, let's pull all of this complex biology together to see the whole picture. We have gone on quite a journey today. We started with the canine distemper virus, a sneaky negative sense morbillivirus that enters through the respiratory tract.
Right. It slaughters the CD4 plus T-cell generals to completely crash the systemic immune system.
And then it rides inside circulating white blood cells or through the cerebrospinal fluid directly past the high-security tight junctions of the blood-brain barrier.
Once inside, it initiates a silent invasion during the immunosuppressed window. It causes a restricted infection in the oligodendrocytes, dropping their hacked RNA blueprints onto the factory floor, halting the transcription of the PLP myelin gene without actually killing the cell immediately.
And simultaneously, it begins disrupting the internal transport mechanisms of the nerve fibers, rotting the axons from the inside out.
And then, the tragic irony. The dog's immune system finally recovers, wakes up, and rushes the brain. And in its frantic, dysregulated attempt to kill the hidden virus, it causes massive bystander demyelination.
We see a cytokine storm driven by TNF-alpha, a flood of active MMP enzymes dissolving the extracellular matrix, and macrophages literally rusting the iron-rich myelin factories to death with violent bursts of oxygen radicals,
Leaving the surviving dog with chronic, progressive, seemingly permanent neurological deficits because the mutated virus goes into stealth mode, hiding its fusion proteins and passing silently cell to cell, completely evading the antibodies.
Until we introduce the epidural delivery of mesenchymal stromal cells. By injecting them into the spine, we bypass the pulmonary trap in the lungs.
The MSCs use their specific receptors to follow the chemokine breadcrumb trail directly to the demyelinating lesions.
They act as biological foremen, flipping the local microglia from a neurotoxic M1 state to a healing M2 state, and pumping out BDNF and VEGF to regrow the axons, stimulate the local progenitor cells, and lay down new myelin.
And 3 doses later, over the course of 90 days, paralyzed dogs are standing up and walking.
It is the very definition, the highest realization of regenerative medicine.
It really is.
It is breathtaking science. But as we wrap this up, it forces me to think about who else this matters for. Because if you are listening to this, unless you happen to be a veterinary neurologist or a very dedicated dog breeder, you might be wondering why we just spent an hour doing a PhD-level deep dive into a canine virus.
And that raises what is perhaps the most important question of this entire discussion. It is the most provocative, wide-reaching takeaway from this entire stack of research.
Lay it on us. Why does this matter for human medicine?
Well, in biomedical research, we cure horrific diseases in laboratory mice every single day.
Yeah, we're very good at saving mice.
We reverse paralysis in mice, we cure cancer in mice, and the vast majority of the time, those cures completely fail to translate to human clinical trials. A mouse brain is tiny, its immune system is different, its metabolism is completely alien to ours.
But a dog.
A dog's brain is highly comparable in size, complexity, white matter distribution, and physiological metabolism to a human brain.
And canine distemper isn't just some random niche disease.
No, it is not. The specific neuropathology of chronic canine distemper, the viral persistence, the restricted infection of the oligodendrocytes, the massive macrophage-mediated bystander demyelination, the specific imbalance of MMPs and TIMs driving the lesion growth, it is strikingly, almost terrifyingly similar to the pathology of multiple sclerosis in humans.
Wow.
In fact, it also perfectly mimics the pathology of post-measles encephalitis in children.
So the provocative thought we are left with is this. If a rigorously timed epidural protocol of mesenchymal stromal cells, 3 doses, 1 million cells per kilo, spaced 30 days apart, can effectively reverse chronic progressive demyelination in the complex brain of a dog.
A condition the entire world previously considered permanently irreversible.
Could this exact cellular therapy protocol, this specific epidural methodology, be the missing blueprint for curing multiple sclerosis in humans? Are we looking not just at a veterinary triumph, but at the direct, highly translational foundation for human clinical trial?
The lines between veterinary miracles and the future of human regenerative medicine are getting incredibly wonderfully blurred.
It makes you realize that maybe, just maybe, the diagnostic X-ray machine for demyelinating diseases isn't permanently broken after all. Maybe we just needed a completely different kind of film, a cellular film to finally see the cure. Something to mull over until next time.